Hi, today I'll be talking about this paper titled Light-Regulated Gene Repositioning in Arabidopsis by Dr. Min Chen and et al. So in this paper, they're again talking about light response photomorphogenesis, and particularly about how genes reorganize themselves in response to light. In this case, they talk about certain genes in the plant nucleus that reorganize themselves to the periphery of the nucleus in response to light. So then they first describe a tagging method where they can see where these genes are, and they use something they call FISH, which is basically fluorescence in situ hybridization. They basically just hybridize their gene of interest with some fluorescent labels, but they note that this method doesn't really work well with genes less than 10 kilobase pairs in size. So then they use this method where they amplify the gene of interest into a much longer version of itself, basically a lot of repeating versions of this gene, so that it's a bigger target for their fish sort of tagging here. You see here that they just use a kind of what they call rolling circle amplification, and they then test their product in this next figure. You see in the red is the fluorescence of the gene of interest that they amplified, and in the green is another gene that they know is close to their gene of interest, and they mark these with a different method called co-immunolocalization. This method is a little more certain, and they're basically trying to use this to confirm that their fish tagging method does accurately tag their genes. You see in this merged picture that the green and red overlap, so it's basically a confirmation that fish accurately tags the genes of interest where they're supposed to be. So now that we know that this tagging method works, we can see where the gene of interest lies. This gene of interest is CAB, which expresses certain genes in the chloroplast, and it only activates itself when the plant is exposed to light for the first time. So we see in the dark stage that this gene is hidden more in the center of the nucleus, and in far red or light, it moves itself way to the edge of the nucleus, confirming that certain genes do relocate themselves. On the bottom here, we see the transcript level. In far red or red light, the expression of CAB is much higher. It's interesting to note that in kind of like the time-lapse exposure to light, the transcript level is much lower than in the immediate exposure, whereas the the CAB locus itself is still at the edge of the nucleus, but the transcript level is kind of lagging behind. And so they basically reason that the transcription of the gene and the relocation of the gene happen at their own separate paces. This figure here is just kind of confirming that this phenomenon happens with a whole bunch of genes, not just CAB. Here we have RBCS1A, or GUN5, or PC, and all of these rather important genes all kind of exhibit the same phenomenon, where in the presence of red light, they get transcribed and they relocate themselves to the periphery of the nucleus. And in this figure, it's kind of just confirming, once again, the importance of phytochrome, where if we mutate phytochrome, we don't see what we originally saw. So it's really important to have phytochrome in the picture. These genes don't directly themselves respond to light. They respond to the activated phytochrome. Phytochrome is the guy that senses light in the first place. So now that we know that these genes do relocate themselves and transcribe themselves in the presence of light, they're trying to find what genes kind of repress them in the first place. Here in this graph, we can see some of their candidate genes, such as COP1, DET1, and then PIF. And then they mutated these genes and see what the effect was. Inside the darkness, we see some of CAB expressing itself, whereas normally it wouldn't express itself once we have these mutated genes, we do see some expression. So it basically proves that these genes here are responsible for keeping CAB repressed uh, when it's in when it is in darkness, and when it's exposed to light, they kind of get degraded, so the CAB will relocate itself to the periphery and it get expressed. It's interesting to note that some of these genes seem to repress CAB more than others, such as COP1 or DET1. Basically, the reason that these two genes have kind of like a dual function where they both repress CAB from being expressed, but they also help transcribe it once CAB is being activated. So if you take them away, you take away the repression, but you also take away some of the expression. So that's why we see that they don't express CAB as much when we take them away, because they kind of have this dual function here. And ultimately, they paint this picture of how the whole thing works. They have phytochrome at the top, and phytochrome repressed kind of keeps all of these repressors in the way. Once it's activated, it's let goes of its hands, and then these repressors kind of let go of the CAB gene. It just naturally repositions itself to the nucleus, to the edge of the nucleus, and it gets fully transcribed. And basically, the importance of this paper is kind of really highlighting the role of repositioning, where a lot of genes reposition themselves at important stages of biological change to get better expressed, to get better transcribed, kind of get more of the limelight, so to speak, inside the biology of the organism. And that is all for this paper.